We use antibiotics a lot in the lab as selectable markers to select for or only allow to grow bacteria containing a plasmid of interest. So a plasmid is a circular piece of DNA. We can design these plasmids so that they contain genetic instructions to get the bacteria to do things that we want, such as make lots of copies of that DNA, or even to make protein based on the instructions of the inserted DNA that we put into that plasmid. So in addition to our inserts, the stuff like the unique information that we want this bacteria to make for us, we also include a selectable marker, and typically this is an antibiotic resistance gene. This way, the bacteria, if they have that plasmid, they'll also make this antibiotic resistance product. Now, if we grow the bacteria in the presence of that antibiotic, only the cells that contain the plasmid, and therefore hopefully they also contain that insert that we put in there, they'll be able to grow, whereas other bacteria, such as cells that didn't take in that plasmid, and other bacteria that just like wandered in there won't be able to grow. And this way, we're only going to be feeding those bacteria that are actually working for us. And so we call this antibiotic-based selection. Some of the antibiotics we commonly use for this are ampicillin, streptomycin, kanamycin, tetramycin. And so today I just wanted to give some practical details about how we go about using these antibiotics for selection in the lab. Things like how you make stock solutions, when you add these antibiotics right before you use them, and things like that, as well as some of the more theoretical details as well in background as to how these antibiotics are working, not just because it's cool to know, but also because it helps with the practical things in the lab, like how long do you need to allow these cells to recover, and when do you need to watch out for things like satellite colonies, which are those little colonies that are around the bigger colony that don't actually contain what you want. They've just been protected by that bigger colony, such as if they were using ampicillin, where the antibiotic resistance product gets secreted from those the cells that actually have it to kind of protect the cells around it, which can be an issue. So lots of different practical details of using these antibiotic selection markers in the lab, as well as the theory of how it works. So let's dive in.
This will be our rubber plate. So basically there's this bacteria food in this gel, and then we can grow bacteria on top of it. Or we could also be growing bacteria in suspension, and in both of these cases we use antibiotics. What's nice about when you're plating it is that you're going to be able to get these distinct colonies where basically each of these spots is going to represent a single genetically unique population of bacteria, at least in theory. In reality, there can be little variations, so it might not just be all exactly the same bacteria. And so if you really, really want a pure population, you'll have to then take a colony and like streak it and then new colonies and things like that. And even that might be not be totally perfect, because bacteria are going to be evolving and generating mutations randomly and all this various stuff. But typically we just take it for granted that one of these colonies is going to be a genetically unique population of bacteria. Basically, one of these bacteria is going to take root and then kind of just grow and multiply on top of itself so that you get one of these loopy colonies. And what's really important is that you have these distinct colonies, and you can see that there's nothing else growing around it. And that's because this plate is spiked with antibiotics that contain the antibiotic that is resistant, that the bacteria that have the plasmid is resistant to. So a plasmid is just this extracellular piece of DNA that the bacteria can host in addition to their own genome, their own genetic information. So they have their own bacterial genome, their own chromosome, and then they also have this plasmid. So typically this plasmid is like a circular piece of DNA that the bacteria can host outside of their own DNA. And so bacteria can naturally host plasmid, and this can actually be a problem with antibacterial resistance in the wild. But it's really helpful for us in the lab because we can use molecular cloning techniques to actually design these plasmids so that, say, we can stick the genetic instructions for making a specific protein into a plasmid, stick that plasmid into bacteria, and get the bacteria to make the protein for us. We want to make sure that we only that the bacteria that we're feeding have the plasmid that they're going to make the stuff for us. If we just let any bacteria grow, well, in addition to the bacteria that just kind of like wander in there because we don't have a perfect sterile technique, There's also going to be bacteria that either didn't take in that plasmid in the first place when you tried to put it in there through a process called transformation, or they did take it in there, but then they lost it. They lost that plasmid, or they lost the um, antibiotic resistance to some mutation. Because what happens is that having this plasmid and having a resistance gene, it can actually be a fitness cost for the bacteria. So they have to, every time they replicate, they have to spend energy to replicate that plasmid. And because antibiotics typically target key bacterial processes, in order to get around the effects of these antibiotics, bacteria often have to change important things about themselves that might give them a growth disadvantage. And so by doing so, if the antibiotic is not around, these bacteria might die out or they might lose that antibiotic resistance. And so it's important that you include the antibiotics so that those cells will have a growth advantage. The other cells will get killed off, or at least they'll stop growing, and your cells will proliferate. In addition to the plasmid that you design and put in there, there might be additional plasmids in the cell. So for example, we can use specialized expression strains. So if we want to make a protein, we can use strains that have basically these plasmids containing rare codons. So basically the instructions for protein letters are given in the RNA form. They're in like these 3-letter words. These 3 RNA letters spell one protein letter basically. And then these tRNAs come around and add the amino acid letter. Much more on this in other posts, but the basic thing to know is that basically cells will stock up in different amounts of these different tRNAs. And you can basically, if the sequence uses a bunch of these needs a bunch of these tRNAs that the bacteria doesn't have a ton of, well, then you might want to add extra ones in, and these extra copies of these tRNAs can be added on these plasmids that are already in those cells. And then you put your own plasmid into with instructions for making a gene. And so you're going to have to add a second antibiotic in order to select for the cells that actually have the rare plasmid genes. And so you can see on these plasmid maps, when you look up a plasmid, you'll see something like this where it has the various genes, and also you'll see some sort of resistance gene. So in this case, it has kanR, which is going to provide resistance to chloramphenicol. So when you see those little R's, it's typically representing resistance. And you can see that this has kanR, this one has strep-spec, so it provides resistance to streptomycin and spectinomycin. And so we'll talk more about these different antibiotics later. But before we go into those details, it's important to note too what antibiotic selection can and can't tell you. So when you design a plasmid, you typically design it so that the antibiotic selection marker is is on the plasmid that has your insert. But during the cloning process, typically you're just putting in this insert into the same plasmid. You might even be cloning something out of one and putting your insert in. The antibiotic selection marker is only going to tell you that the plasmid is in there. It's not going to tell you if the insert is in there. And so in order to tell you if the insert is in there, you're going to need to do things like analytical restriction digest, where basically you try to cut it and see if it's in there, or colony PCR, where you try to amplify that region that has the gene and see if the region really is there. Or you can do things like to know for sure and to know that there's no typos or anything, you send it for sequencing to make sure that the sequence is in there. So the so the selection marker is only going to tell you about the presence of the plasmid and not the presence of the insert. And so this is going to be an important thing to keep in mind. Another important thing just a notation thing, is that when we talk about selection, this is in contrast to screening. So selection is where you're actually just like only allowing things to grow that have what you want. We can also use a method called screening in the lab, such as blue-white screening. In this case, the things that don't have what you want and the things that do have what you want are different colors, but you're not removing anything. You're just kind of like showing the differences. And so this is another common method that we use. In this case, it can tell us about the presence or absence of an insert. So I'm using the word insert instead of like gene because basically it doesn't have to be a gene. And in technical terms, the gene is actually the DNA version of the of the instructions for making something. So for making a protein, or even for just making a functional RNA. So basically an RNA that acts by itself. A gene is a DNA version, and so what we want to put, a DNA gets made into a messenger RNA copy, which is kind of like an edited version of that DNA, which then gets used by the ribosomes to make the protein. Because this DNA is edited, it gets kind of like spliced together, where the regulatory regions get cut out, and the coding regions, the regions that have the instructions for making the protein, get stitched together. And so this edited version, you want to put a complementary DNA, so a cDNA version of that into the plasmid. And so typically the insert that you're putting in is actually going to be a cDNA rather than a gene. It's easier to talk in terms of gene, but it's not like just in terms of, day to day we might say gene, but it's not actually the gene that you're putting in. Additionally, because you could be just putting in something like, if you want the bacteria to just make a lot of copies of the DNA, it doesn't even have to be a cDNA you're putting in there. And so insert is the most overarching umbrella term. And so we have in our plasmid, we have our insert, and then we also have our selection marker. And so this would be typically the antibiotic resistance gene. There are a bunch of other features of various plasmids, and you can learn about these more in other posts too. So it's not just the antibiotic that you care about, but the antibiotic is one of the key things that you will care about. If you're using a plasmid, it's important that you know what the antibiotic resistance gene is. You can often find plasmids containing inserts of interest on repositories such as AdGene or DNA-SU, and more on these in other posts. But you'll typically get some information about the plasmid that you look up, including the antibiotic resistance. So a common plasmid that's used for expression is like this PET system. And you can see here that some of these have ampicillin resistance, some of these have kanamycin resistance, and you'll see other sorts of resistance genes in other plasmids. So note on naming, so the antibiotics are usually naturally produced by other bacteria in order to protect themselves. So that's where we get these issues where bacteria can actually share these resistance products with one another, because some of the bacteria have evolved to have resistance to the antibiotics that are produced by bacteria that live around them. And then if they share that resistance gene with another bacteria, well, that can be an issue. But in the lab, we can take advantage of these different genes and use them. And we also give them a little credit by naming them with an homage to where they came from. So the resistance genes that come from Streptomyces bacteria normally get names ending in mycin, so this would be like streptomycin, kanamycin. And those from Micromonospora bacteria get names ending in mycin, so gentamicin, or I don't even know if I'm pronouncing it right, but with an I as opposed to with a Y. When we use them in the lab, there are some practical things to know. So in terms of how we're growing bacteria, as I mentioned, we can be growing them kind of like on these petri dishes, these LBR agar plates, where LB stands for lysogeny broth. It's a basic bacteria food that we use. There are other types of bacteria food that you can use too, and that you can make in these plates, because basically it's just this agar, so it's like this sugar that we mix with this bacteria food that we can make this liquid food, and then mix it in with that, and then make these plates. And we'll talk about that more in a minute. But we can grow them on these plates, so we can grow them in this liquid. But in either case, it's really important that we add the antibiotics just right before we go to use them, so that we avoid them degrading before they can actually go to work. And so typically what we do is we make high concentration stock solutions of the antibiotic. So a stock solution is basically just a version of what you want to use that's way more concentrated than you would actually want to use it at. And this way you can add just a small amount of it, and therefore you don't have to, you have plenty of room for other things. So if you were, say, to make an antibiotic solution that was at the concentration that you wanted to use it at your, what you probably call like the working concentration, the concentration you want to work with it at, well, then you wouldn't have room for anything else. Whereas if you make a high concentration, then you just have to add a little, and then you have plenty of room for everything else. It's convenient to make a 1,000-fold stock solution of these antibiotics. And so 1,000-fold basically is 1,000 times more concentrated than what you want. 1,000 is really convenient because we can basically add microliters per milliliter. So if you have 1,000 times X, you would add 1 microliter per milliliter. And so if you had 1 liter of media, you would add 1 milliliter of your antibiotic. And if you had 1 milliliter of your media, you would add 1 microliter of your antibiotic, but you wouldn't typically be adding that little. You would probably be at like the least would be like 5 mils or something, which would then be 5 microliters. So it's one of the first things you'll do probably if you start getting started in started working in this lab is making a bunch of these stock solutions. So these are some of the common working concentration. These are some of the common stock concentrations that we use of these various antibiotics. So like 100 mg per mil. This would mean that it would be this is 1,000-fold, so it would really be 100 micrograms per milliliter in terms of your final concentration. Note that for some of these, for like tetracycline, you're going to want to make it in 70% ethanol. Tetracycline does not want to dissolve very well. It's also all orangey. But yeah, you'll see it like precipitate out if you try to make it in just pure water, but if you make it 70% ethanol, you'll have more luck. With chloramphenicol, we make it typically in 100% ethanol. With all of these, you're going to want to syringe filter it. So basically, you put this little syringe on the end of it. It has this filter on the end of the syringe. It has a membrane that has really tiny pores, and so we typically use a 0.2 micron membrane, which is going to keep out any bacteria. And then we can filter it into aliquots. And so you want to make these like single-use portions so that, or at least just a couple use portions. This way you don't have to keep freeze-thawing them. So you typically put these in the -20, so keep them nice and frozen, or in the case where it has ethanol, it's not actually going to freeze, but it's still going to be kept really cold. This is going to prevent their degradation, it prevents any enzymes from being active and destroying them. And it's also going to make it so that if you accidentally contaminate one of the tubes, well, now your other tubes are still okay. And so aliquoting, it comes in handy here as well as in other places. And yeah, typically I don't worry too much about exact volumes of these aliquots unless it's something where I know I'm always going to need like a mil or something, and then I would make them all exactly a milliliter. Otherwise, I just kind of like syringe it in there. And yeah, so you might need to let them mix a little while before in order to completely dissolve. But then you make these aliquots and you can store them, and now you have them ready to use. When you go to use them, you want to make sure that you're adding them right before you're going to use them. This is especially important to when you're making like those LB agar plates. What's actually going to happen, or even in the case when you're not, you're actually going to sterilize the media so that you make sure that, because when you're making these mixtures, you're typically just doing it out on your bench, and there could be all sorts of stuff getting in there. And so you want to sterilize it first before you go to use it. And so we use this autoclave, which is like a super high pressure oven or sauna. It's going to sterilize stuff with steam. And so it's going to get really, really hot, which would totally kill your antibiotics, and you don't want that to happen. And so you're going to want to add them before you pour the plates, but after that media comes out of the autoclave. And not just right after it comes out of the autoclave either, you're going to want to wait until it cools down. So typically I let it cool until you can kind of like touch it before pouring the plates. And before you pour the plates, you're going to want to put in the antibiotic and mix it well so that you have, so that the antibiotic is nice and mixed, and then you can pour your plates. In the terms of just the media, so when you're keeping that media out on your shelf or you're waiting till the next day in order to use it, you don't want to add the antibiotic until you're ready to use it. And if you have like a bottle of the media that you just kind of like keep on your bench and then take portions of, don't put the antibiotic into it, just put the portion, just when you take a portion of it, then add the antibiotic to it rather than try to keep this media with the antibiotic in it over time, because that antibiotic will degrade and then it won't be useful. Okay, so that's the basics of working with these antibiotics. So in general, and now let's talk a little bit more about the specifics for the different types of antibiotics. So as I mentioned before, antibiotics can kind of like target various core mechanisms of the bacteria. and then the bacteria can evolve resistance in different ways or acquire resistance in the plasmid through genes that do different things. So some of these genes are going to allow the bacteria to change the antibiotic, modify it with methylation, acetylation, phosphorylation, something like this, so it's not going to bind. Other things are going to basically change the, make it so that the drugs get pumped out. And so these like efflux pumps, things we'll see like TET-A, which will pump out tetracycline. And there are also other ways that the bacteria can kind of get around the effects of the drugs. So one of the common types of drugs that we use are beta-lactams, things like ampicillin and penicillin. What these do is they target the synthesis of the cell wall. So our cells don't have a cell wall, but bacterial cells do, and their cell wall has this peptidoglycan. And peptidoglycan is basically these sugar chains that are linked together by these little peptides, these short chains of amino acids, these kind of like little protein chains that are linking together the sugars. In order to make those chains, the bacteria uses enzymes, so these reaction helpers called transpeptidase, and this is referred to as a penicillin binding protein, or PCB, or PB, I don't know why I did that wrong. But basically what this is going to do is it's going to make those linkages. And in order to make those linkages, it kind of has to attack one of the peptides to link it to the other, and it gets temporarily stuck on that peptide. What happens is that if we use an antibiotic, a beta-lactam antibiotic, where that beta-lactam refers to this ring structure, what happens is that transpeptidase, it goes and it attacks one of the peptides in order to try to link it to the other, but instead of then kind of just doing a pass off, it gets stuck on that first peptide, and this inactivates the transpeptidase, so it can't make the strong cell walls. What can happen is that the bacteria can, if they have this another enzyme, so if they have beta-lactamase, what can happen is that they can inactivate that beta-lactam before it can go and confuse the transpeptidase. So basically, the beta-lactamase is going to attack it and break it open, so that it's no longer going to be able to inhibit the transpeptidase. And so this BLA gene is going to provide resistance to ampicillin. And so this is a common pair that we use for antibacterial resistance selection in the lab. An important thing about using it in the lab too to know is that ampicillin, it's the BLA, this beta-lactamase that it makes, is actually shipped out of the cells. So basically these cells go on the offensive and they ship this antibiotic product out of themselves. This is going to kill the antibiotic before it even gets into the cells. And because it's shipping it out, it's protecting itself, but it's also protecting the things around it. And so what you could end up having is these like satellite colonies, which are these little colonies around that major colony that actually has your plasmid. You get these satellite colonies that don't actually have the plasmid; they're just getting protected by the ampicillin, by the shipping out of that beta-lactamase. This can particularly be a problem if you're overgrowing the cells. So ampicillin, you want to basically don't let them overgrow, so grow them until they're big enough that the colonies are pickable and nice and stuff, but you're not seeing the satellite colonies as much. Just another note, so ampicillin degrades fairly easy. This is why you want to limit the growth period of these of cells in ampicillin, as well as you want to add the media or add the antibiotics to the media right before you use it and not to in advance. There's a version called carbenicillin, which is more resistant to degradation, but is also more expensive, and typically you often use just ampicillin in the lab. We also typically commonly use antibiotics that target the protein making, so target translation. And so basically in the process of translation, these tRNAs are bringing amino acids, so bringing these protein letters to the ribosome, which is the protein making machinery that's going to piece them together based on the instructions of the messenger RNA. Much more on this in other posts, but this ribosome is going to be a key target for many classes of antibiotics. These include the tetracycline antibiotics, and Basically, what happens here is that these antibiotics are going to bind to the ribosome and prevent them from binding to those tRNAs and prevent the bacteria from making proteins. Now, the bacteria can get around this if they have the TET-A gene, which makes an efflux pump. This efflux pump is basically going to pump the drug out of the cells so that the cells are not affected by it. Another antibiotic that targets the ribosome is kanamycin. The same thing goes for other aminoglycosides, things like streptomycin, spectomycin, gentamicin, neomycin. These are going to bind to the ribosome and kind of mess up its ability to proofread, leading the ribosome to make these dysfunctional proteins. The kanamycin resistance gene, so kanR, neo or NPK2, it makes an aminoglycoside phosphotransferase. So basically, these drugs are called aminoglycosides. They have this amino groups and these glycosides, so basically sugary things. And a phosphotransferase is something that transfers the phosphate group, so this negatively charged bulky group, onto it. This is going to make it so that it's not going to bind to the ribosome, and therefore the bacteria is going to make proteins normally, and it's not going to be an issue, and it will survive and thrive. So kanamycin and the aminoglycosides, they're going to basically mess up proofreading, and then you have the transfer, you modify that molecule, it can't bind, all's good. Tetracycline is going to prevent the protein making. Tetracycline A, the TET-A is going to make an efflux pump that's going to pump it out. Another one that targets translation is chloramphenicol. It's also going, this is one of those, remember, that you're going to want to dissolve in ethanol. One of the cool things about chloramphenicol is we can actually use it to increase the plasmid copy numbers, or how many copies of the plasmid there are in the cells. So the cells can actually host multiple copies of a plasmid, and you can kind of use chloramphenicol in order to control how many copies there are of certain plasmids. What happens is, because chloramphenicol is blocking translation, but it's not blocking the actual, it's not actually killing the cells. It's what we call bactericidal. It's what we call bacteriostatic. So it slows the bacteria down, but doesn't kill it. So if you target the amount of antibiotics that you add, so that the cells are switching to kind of making copies of the plasmid rather than making protein, you can therefore get them to make more copies of the plasmid. And so there are a couple of different methods. I've probably more in other posts, but you have this Maniatis method, where you grow it to saturation so the bacteria can't grow anymore. There's not enough food or land. And then you add a lot of the chloramphenicol, enough to prevent growth, but they can't grow anymore anyway. And so then they'll just make lots of copies of the plasmid. Or there's the Begmine method, where you just add the chloramphenicol early on, but a lower concentration. And that'll shift the resource allocation towards plasmid copying. This will work only with certain plasmids, the ones that have kind of like this relaxed control, where they don't need to make proteins in order to replicate the plasmid. So some bacteria, some of the plasmids, you need to have it, or some of these. So some of these the bacteria have to make proteins in order to replicate those plasmids. And these are called like stringent control, whereas with relaxed control, they don't need to make any proteins. And so if you need to make proteins, well, that would be an issue, because chloramphenicol is inhibiting the protein making. But if you have one of these relaxed plasmids, then the strategy will work, as long, of course, as your plasmid doesn't have a chloramphenicol resistance gene. And so if they have a chloramphenicol resistance gene, commonly it says chloramphenicol acetyltransferase, or CATS, this is going to inactivate chloramphenicol by acetylating it and preventing it from binding the ribosome. Note too that you'll see a lot of different abbreviations for chloramphenicol. You might see CHL, you might see CM, you might see CAM, CHLO, or various things like that, but those are standing for chloramphenicol. No matter which of these antibiotics you're using, you typically, when you put your plasmid into cells, we call this process called transformation, and a common way that we do this is with heat shock. We can also do this with methods like electroporation, various methods in order to get the plasmid into the cells. Often what we're doing is we're just doing this, when we're doing this transformation, we're doing it in just like a small portion of cells. So we're like in one of those little Eppendorf tubes. And we stick the plasmid into the cells, and then basically, in order to get these cells to take in the plasma, we kind of have to hurt them. So often we make these cells chemically competent, so we weaken the cells in order to weaken their walls and stuff so that it makes it easier for them to take the plasmid in. And so they're going to need to recover afterwards. And so if you want them to recover, you're not going to want to put them in a stressful situation. So you're not going to want to put them in the antibiotics right away. Instead, you typically do an outgrowth step. In this step, you grow it in the absence of antibiotics for like half an hour to an hour, allow it to recover and allow it to start making those antibiotic resistance products so that it can avoid the effects of antibiotics before it gets killed off. So how long you let them recover is going to depend in part on the antibiotics that you use. In order to heal itself, the bacteria is going to have to make some proteins. Now, if your antibiotic is inhibiting protein making, that's going to make it so that it's harder for the bacteria to heal themselves. And so if you're using an antibiotic like kanamycin or tetramycin, if you're using an antibiotic like kanamycin or tetracycline or chloramphenicol, something that's inhibiting the protein making, well, then you're going to want to wait, give it an outgrowth of about an hour in order to allow it to make the proteins that it needs to heal itself, as well as to make the proteins to inhibit the antibiotics when you add them. Alternatively, if you're using something like ampicillin, well, here you're targeting the cell wall, not the protein making, so it'll still be able to make those proteins. And so you don't need to give it as long as an outgrowth. So typically I'll give it like half an hour. At that point, you can then go plate your cells, stick it in the static incubator with the plate upside down so that any condensation is falling on the lid and not onto your plate. And then the next day, hopefully you'll see some colonies, and you don't want to leave your plate in there too long. You want to leave it in there so that you have these nice biggish colonies, but they're not getting overgrown and you're not starting to see the satellite colonies, because remember, the antibiotic is going to be degrading in these plates. And so typically too, when you have your plates containing antibiotics, you're going to want to keep these in the fridge. So say in one of those deli fridges, if you have one of those are really nice because you can keep these stacks of plates. But that's the basics of working with antibiotics. So the key concept is that you have the antibiotic resistance gene on the same plasmid as the insert, so of the thing that you want the bacteria to be making copies of or making protein from, various things like that. The antibiotic resistance gene is going to select for the piece of DNA that's holding them both. And in this way, you're going to be able to allow it to have a growth advantage, whereas it would normally have a growth disadvantage because you're making it do more work. But this way, it'll have an advantage over those other cells. It'll be able to outcompete them. And then you'll end up with, as long as you keep using the antibiotic, you'll end up with consistent population that has your product of your thing of interest. Remember though that you have to keep using this antibiotic, or else the cells can lose that plasmid because it will probably be a fitness disadvantage. So you have to use the antibiotic throughout whenever you're working with these bacteria. So hope that helped.